for us. Dr. Issabelle Ampofo recently defended her PhD in animal genomics and genetics at the University of Connecticut. Her research spans genomic selection and diversity management in aquaculture and livestock, with applications to disease resistance in Pacific white shrimp and crossbred evaluations in dairy cattle. She integrates SSG blood, metafounders, and population genomic analysis to improve accuracy while safeguarding long-term diversity. Issabelle completed a quantitative genetics internship at ZOED, has presented at major conference, and served as a TA for Principles of Biology. So I'll turn it over to now Dr. Issabelle and can get started.
Thank you. Hi, thank you so much for inviting me to give this talk. So for today, I will be presenting some results that we have from my PhD studies at the University of Connecticut. And before I begin, I wanted to talk a little bit about myself. So as rightly said, I'm Issabelle Ampofo. I was born and raised in the capital of Ghana. It's called Accra. And I lived there all my life. Then in 2014, I went to the University of Cape Coast where I got my bachelor's in molecular biology and biotechnology. And then during COVID in 2020, I was awarded the Erasmus Mundus Scholarship in animal breeding and genetics. And so I moved to the Netherlands, specifically to Wageningen, where I started my master's in animal breeding and genetics. Because it was a dual master's program, I also had to finish my last year at the University of Natural Resources and Life Sciences. And I had the opportunity to work with really great scientists, which included Peter Beimer and Johann Sockner that you see over here. And then in that same year, 2022, I was very privileged to have been interviewed by Breno Fragomeni, who is here, for a PhD position in shrimp genetics. And so then I moved to the US in 2022 and I've been there till now. Last week on Friday, I defended my PhD dissertation and I successfully passed. So as rightly said, I'm a doctor now. And yeah, that's a little bit about me and my background. So I'm going to dive a little bit into the background study for the presentation that I will do today. And we will first look at how the production of L. vannamei works like. So basically, the first spawning of this species was first recorded in 1971 in Florida. And this was from a Naupoli that was spawned from a wild female that was caught and shipped from Panama. Now, following the development of intensive rearing and breeding techniques, we have commercial cultures of these species available everywhere. As of 2021, the global shrimp market by value was estimated to be around 31 billion US dollars. And this number is expected to increase to about 74 billion US dollars by 2028, making L. vannamei the most commercially important shrimp species. As we speak today, the United States stands as the largest importer of farm shrimp. But irrespective of the profits that have been made in this industry, several diseases continue to pose a threat. And one of such diseases is what we'll call the acute hepatopancreatic necrosis disease. It is a bacterial disease that is known to be caused by binary toxins produced from specific strains of the bacteria Vibrio parahemolyticus. Now, Vibrio parahemolyticus is said to be a gram-negative bacteria that is usually found in microflora of aquatic environment. And in regions where the disease is said to be exotic, on-farm prevalence is usually between 40 to 100%, with mortalities seen as early as within 10 days when ponds or farm water is stocked with the post-larvae or the juvenile shrimp. The main mode of transmission is usually through the ingestion of bacterium colonized feed pellets and water, and also through the cannibalism of sick or dead shrimps by other shrimps. Now, if you take a look at this picture that I have here, we see a normal shrimp. And in that shrimp, we see that the stomach is full, the hepatopancreas is brown in color as it should be, and the gut is full. But in the case the shrimp experiences the disease, symptoms will include seeing an empty stomach, a pale or atrophied hepatopancreas, which is due to the degradation of epithelial cells within the hepatopancreas, and then also an empty gut. As of 2019, the cumulative losses from the disease was estimated to be around $9 billion. And even though antibiotics have been used as an option to mitigate the effects of the disease, it has somewhat been limiting due to the fact of concerns of environmental pollutions in open systems, and also due to concerns of rise in antibiotic resistant strains of bacteria. Now, vaccine development has also not been feasible due to the fact that when you take a look at the innate system of the shrimp, it lacks a memory, which is very important for the production of antibodies. Therefore, we believe that there is a need for an approach that is more environmentally sustainable and friendly to help mitigate the effects of these diseases. And from an animal breeder's perspective, one of such alternative would be to breed for disease resistance, because breeding for disease resistance could help farmers to cope with the adverse losses that are associated with this disease. And even more so, with the availability of single nucleotide polymorphism, genomic selection has become quite popular, especially for traits that are hard to measure, and for traits that are destructive to measure, and for traits that we cannot measure early in life for the individuals and the study. But then when it comes to genetic gain, even though we have a lot of selection tools right now that helps us to accelerate the rate of genetic gain, we know that in the process of selective breeding, it can lead to a reduction in genetic variation within a selected population. We know that it can lead to an increase in autozygosity, as we would also call as inbreeding, which we can also talk about it in terms of a decrease in the phenotypic performance of a trait of interest. And then we can also talk about it that it could also lead to a decrease in the effective population size of the population you're trying to improve. And so we believe that we should be able to set up guardrails that will be able to help us monitor these diversities while also maintaining long-term genetic gains in a selected population. So that brought us to our long-term goal, which was to implement a genomic selection scheme for resistance to AHPND in a commercial population of L. vannamei and the challenge conditions. And we had so many objectives in order for us to meet this long-term goal. But for today, I'm just going to look at two of the objectives that we set out to look at. So for the first one, it was to investigate the genetic basis of response to AHPND in two commercial lines of L. vannamei and the challenge conditions. And in order to do this, we wanted to estimate genetic parameters for survival, compare different validation approaches for prediction accuracies, and also look for associated loci in a genome-wide association study. For our second objective, it was to characterize the genomic diversity and also look at the recent demography in these two lines of L. vannamei that are undergoing selective breeding. And in order to do that, we were to estimate genetic diversity indices that included observed and expected heterozygosity. We also evaluated the population structure and also quantified genetic differentiation between the lines that we were looking at. We quantified linkage disequilibrium, which is a very important factor for genomic selection, and also we inferred recent effective population size. And then finally, we were able to estimate inbreeding measures from the concept of ranks of homozygosity. So to the first objective, which was to investigate the genetic basis of response to AHPND, we first had to conduct the challenge experiment. And so we started off by selecting individuals from two commercial lines, which are the fast growth line and then the slow growth line. We selected 40 families, that is 20 families from each of this line, and then these animals were then bred under biosecurity conditions at the shrimp improvement systems in Florida. And then when they were about 29 days old, we infected them with about 1.0 times 10 to the power 5 colony forming units of the bacteria. In a second challenge that we conducted in 2023, we looked at the progenies of siblings of the individuals in P2022. We selected the same number of individuals and then we challenged them again. But this time we increased the dose of the bacteria in order to be able to meet the LD50 threshold. And then by the end of those two challenges, we had about 1,999 streams from the P2022 challenge, and then about 2,353 streams from the P2023 challenge. I want you to take a very good look at the numbers we have here, that we had more individuals in our P2023 challenge compared to our P2022 challenge, because this will come back to affect some of the results that we had for our validation strategies. Now, all of these animals were then genotyped with a 50K SNP chip that was provided to us by the Center for Aquaculture Technology. So during the P2022 challenge, we had other phenotypes that we collected, and this included maturity entry rate, which is the weight of individual families at the time of transfer between certain raceway tanks that we had at SIS. We also had another phenotype we called average daily gain in cages, and then the difference between a raceway 8G and 8A goat sampling weight. And then finally, we also recorded bacterial load, which we quantified as the log transform copy number of the AHPND toxin gene, the PAB toxin. So for our prediction models, we decided to analyze survival as a binary trait. And so we relied on a threshold model. So here in the threshold model, we are looking at the liabilities, and even though these liabilities cannot be observed with the naked eye, we know that they can be affected by both fixed and random effects. And so we decided to analyze different models and compare them in terms of accuracies and also inflations of GEVVs. So for the first model, which we'll call model 1A, it was a multi-line model. So it included 2 lines, and then we only included RAC and then the challenge number as fixed effects while treating the animal as a random effect. And then in a separate model that we will call 1B, it was similar to model 1A, but in this model, analysis were done separately within each line. So it was a single line analysis, and this was done in order to account for potential line specific effects. And then we also had a model we would call model 2, and in this model we just expanded upon model 1A by including line as an additional fixed effect alongside RAC and challenge, and we still maintain animal as a random effect. And then finally, we wanted to address the problem of population structure, because we have 2 lines that we are combining in a single evaluation. And so we decided to include the first 3 principal components as covariates within the model. And so here we will term that model as model 3. And in order to validate our prediction models, we decided to use 3 approaches. So in the first approach, we will call the forward in time prediction. And in this approach, the animals in P2022 served as the training set, and then the animals in P2023 served as our validation set. So we were using the predictions made from we were using the GEVVs made from P2022 to validate the animals in P2023. And then we also did a backward in time prediction. This was the reciprocal or the vice versa of the forward in time. So here, animals in P2023 rather served as our training population for animals in P2022. And then finally, we had a K-means clustering. Here, you're looking at the number of clusters that you have with that are statistically significant within your population, and then it's just like a cross validation where each cluster is going to serve as a validation set at least once. And then to compare these validation approaches, we had 2 matrices. So we're looking at accuracy of predictions, and we're also looking at inflation. So here we calculated accuracy as the square root of the covariance between GEVVs that were calculated with a complete data set and GEVVs calculated with a reduced data set, and then we divided that by the genetic variance that we estimated from the trait. And then we also had inflation, which we will describe here as the slope of the regression of GEVV from a complete data set on GEVVs on a reduced data set. And the expectation here is 1, so that if you have a value that is greater than 1, we can say that the behavior of your GEVVs is such that they are underestimated. And if you have a value below 1, then we can say that your GEVVs are overestimated. And next to our genome-wide association studies, we decided to keep model 3, which was a multi-line model in order to combine the two lines. And then for a single line approach, we relied on model 1B. So after solving for the GEBVs using Gibbs F90 plus, we back solved the GEBVs to SNP effects using post GS F90. And then we decided to use the sliding window approach to look at the percentage of variance explained by at least 0.5 MB. That was the threshold we used. We also looked at 1 MB and 0.1 MB, but in the peaks we had from all of that were similar to the 0.5 MB window variance that we chose. So to some results, first of all, I'm going to show the survival descriptives that we had from the P2022 challenge. And as we can see here, the red bars represents the families in the fast growth line, and then the blue lines represents the families in the slow growth line. On the y-axis, you have the survival percentage of these families, and on the x-axis, you have the family numbers. And as you can see right from the plot, the fast growth line had a higher survival percentage compared to the slow growth line. We had about 60% on average survival in the fast growth line compared to about 30% in the slow growth line. But then when we go to P2023, we saw something different, that the survival percentage of the fast growth line families decreased drastically. And then we had just a little improvement in the slow growth line. So even though we saw these differences within line, we also saw that there were differences within the families, and we think that we can exploit that variation for our selective breeding. But then when it comes to the variations that we see phenotypically, we also wanted to see how much of that is quantified genotypically. And so we relied on the concept of heritability, here explaining the amount of variation that you see in the trait that is attributed to genetics. So these are the models that I've already talked about. And then we see that for the single line analysis, which is model 1B, we had for fast growth line about 0.52, and then for the slow growth line 0.39. But then across all models, we see that heritability was really high, ranging from medium to very high, showing that selecting for there is enough genetic variation in the traits for survival that we think we can select upon to improve animals. When it comes to prediction accuracy, so on the x-axis here, we see the models that we analyze, and on the y-axis, we see the accuracies. And then the different colors represent the different lines together with the kind of validation that was done. And so you have the green line representing the fast growth line in a backward validation, the orange representing slow growth line in backward, blue representing fast growth line for forward, and then the purple representing the slow growth line in forward. And as we can see across all models, we see that the backward in time prediction gave us better estimates in terms of predictions compared to the forward in time predictions. And then we also saw that for fast growth line, we had our highest accuracies for the fast growth line families compared to the slow growth line families. When it comes to prediction bias, which we measured as inflation, we saw that for the backward in time prediction, we had better inflations on our of our GEBVs. That is, we had values that were approaching 1. So that we see that for the backward in time, especially for the fast growth line, we see values that were approaching close 1. And then especially for model 1A and then model 3. But then the slow growth line showed severe overestimation of GEBVs across all models. When it comes to claimings clustering, where we only applied it to model 3, when we did the total sum of squared by comparing it to the number of clusters, we saw that the number of clusters in our population, the optimum number of clusters in our population was 3. And out of these 3, 2 of them were identified within the fast growth line, and then 1 of them was identified within the slow growth line. And so next we did the k-means cross validation, and we see that across all clusters, we had very severe underestimation of our GEBVs, like values that were more than one, and then our accuracies were really low. And this makes sense because we have two distinct lines, and we are trying to use the predictions of one line to validate the predictions in another line. And this is the problem that we would usually have with multi-populations predictions. So we also wanted to see if there were any potential trade-offs between survival and then other traits that we had measured in the P2022 challenge. And here we see that for survival against growth, we saw a high positive correlation, which tells us that possibly if we select for survival to AHPN, we may also improve the growth of the animal and vice versa. We also saw a moderate negative correlation between survival, growth, and bacterial load, which also tells us that bigger animals tend to have lower concentration of the bacteria and vice versa. And so possibly selecting for growth may improve AHPN by reducing the bacterial loads in those animals. So this was a very important results that we had from our studies. And then for our GWAS, in our multi-line, fast growth line, and slow growth line, we had about 29 windows that were above, that were explaining more than 0.1% of the variance in our trait. But then we did see that there were two windows on LG17 and LG10 that were consistently identified both across the multi-line evaluation and then the slow growth GWAS analysis. So we did a functional annotation of the genes surrounding these areas, and then we saw that most of the genes were involved in metabolism and enzyme production, also in transcription and regulation, signal and transport, immune and defense, which is very important, and also in cytoskeletal and structural function. So for objective 1, we did see that indeed there is significant survival differences that we observed between the lines, and survival to AHPN is highly heritable, meaning that both lines can respond to selection successfully. We also saw that the prediction accuracies that we had varied by the validation strategy, by the model, and also by the line that we were analyzing. And then possibly selecting for growth may also enhance AHPN survival. And then finally, the GWAS that we conducted did reveal window loci that were linked to cell structure, barrier integrity, and also in immune defense. So before I move on to the second objective, I just wanted to talk about a little fun fact that we know about shrimp that I wanted to share to you. So I don't know if you know that shrimps exhibit sexual dimorphism. So there are some shrimps that will start off as males when they are giving birth to, and then somewhere within their life they would basically switch their sexes from being males to females, which is actually a really cool and fascinating fact that I wanted to share with everyone here. So the next time you see a shrimp, probably you might have a shrimp with two genitals, or a shrimp that has just transformed itself to being a female. So you just watch out for that. Anyway, so to our second objective, which was to characterize genomic diversity and also recent demography in the two lines, we decided to use the genotyped animals that we had from our challenge experiment, and the genotypic data underwent quality control by excluding SNPs and individuals with a core rate less than 0.8, and also SNPs with a minor allele frequency less than 0.05. So that at the end of the day, we had about 2,101 shrimp for the fast growth line and 2,251 from the slow growth line. And in order to estimate genetic diversity, we relied on looking at the observed heterozygosity, the expected heterozygosity, which will be the expected genetic diversity of your population if it was in a Hardy-Weinberg equilibrium. And then we also looked at the average relationship among individuals in the population, which we'll refer to here as pi hat. And these estimations were done using Planck. And then for population structure and differentiation, we looked at the principal component analysis using Planck, and we also estimated fixation index. And then in order to look at how the value we got for fixation index was partitioned between and within lines, we decided to look at analysis of molecular variants, which was done using Adigenet and Pega softwares in R. And then we also estimated LD decay via Plink and also effective population size via the GON software. And then finally, we looked at inbreeding coefficients based on runs of homozygosity by looking at different FRO classes, which included a 0.5 to 1 MB, 1 to 3 NB, and then anything greater than 5 NB. And this was done using the R0ROH software with a mixed KR model. So to some results, we saw that on average, we had observed heterozygosity of 0.28 and 0.31 for the slow growth line and the fast growth line respectively. And those values were a bit higher than the expected heterozygosities in our population, which tells us that we have excess heterozygosity in our population, and that was a good thing for us. And then we looked at the fixation index value, which was 0.21, and based on what Veer and Cockram has spoken about with respect to fixation index, this is a very high value to say that our populations are genetically distinct from one another. Now we wanted to see how this value, the FSD, is partitioned within and between these two populations. And so we went ahead and then we looked at analysis of molecular variants, and we saw that between populations we have about 24% of our variation between population, and most of our variation was within population, which was 75.58. And then if we just look at the sum of squares for the variance components, if you take the ratio of the between and then the within, you get a value of 0.24, which was close to the value of 0.21 that we had from Plink, which again suggests that our two populations are genetically distinct from each other and they have no overlapping ancestry. So for LD, as expected, we saw that when you move away from the farther you are from the genome, the lower your LD is, which is what we saw over here. We saw a mean LD of 0.11 for the slow growth line and a mean LD of 0.09 for the fast growth line. So on average, we have a little bit higher LD in the slow growth line compared to the fast growth line. And then for effective population size, we saw that across generations from 200 years ago, the effective population size has remained consistent up until somewhere 10 years ago, where it dropped sharply. And this is consistent with how the Shrimp Improvement Systems company in Florida has been operating, because they started really selecting for this line about 25 years ago. So it kind of matches the timeline and what the plot we see here. But on average, when we look at the geometric mean of the last 5 generations between both lines, we have an average contemporary NE for FG to be 105, and then for the slow growth line it was around 80, again telling us that the slow growth line may have a smaller effective population size, which is consistent with the large LD that we saw in the previous plots. And if we compare that to the fast growth line, we have a higher effective population size, which is also consistent with the lower LD that we saw in the previous plots. So we also wanted to look at inbreeding across the entire genome. And this inbreeding was estimated based on homozygous by descent from the package that I talked about. In the fast growth line, we had a mean FHBD of about 0.06, and in the slow growth line, it was 0.09, again consistent with its low effective population size and higher LD. But overall, both inbreeding coefficients approached 0, which tells us that we have enough genetic diversity, and probably that's why our observed heterozygosity was a little bit higher than our expected heterozygosity that we saw from the genetic diversity indices. And then also we looked at the different classes of ROH that we decided to estimate inbreeding from. So we see that most of them were approaching zero. The mean inbreeding was approaching zero. However, most of the contributing factor in our populations were from very short segments of our genome, so that you have about the 0.5 to 1 MB contributing most to the inbreeding that we have in both populations. So basically what we see here is that we have very old inbreeding in our population, and SIS is doing a good job in managing recent inbreeding by trying not to mate closely related individuals in recent generations. And then we also did an inbreeding enrichment study where we looked at um, the number of ROH on each chromosome, and then we standardized it, and then we looked at peaks on the we compared peaks on the chromosome to tell us if there's anything happening somewhere that may be a hotspot for either a selection signature or something. And so we saw that on chromosome 7 in the fast growth line, we had a ratio that was a bit higher than what you would have on the entire chromosome, and that is what gave us our largest stretch of HBD, which was between 3 to 5 MB. We did the same for the slow growth line, and we saw the same region on chromosome 7, and it was even higher for the slow growth line compared to the fast growth line. So there might be probably something going on chromosome 7 that I believe that doing a selective signature study may be able to reveal more about. So in conclusion, for the second objective, indeed, we did see that there was substantial genetic differentiation between the two lines with an FST of 0.21, which shows high genetic differentiation. The recent effective population size was between 80 to 105, that is 80 for the slow growth line and 105 for the fast growth line, which is indicative of reasonably high diversity. We had very low inbreeding coefficients in both lines, and most of the contributing segments to this inbreeding coefficients were from very short segments, which is indicative of old inbreeding within the population. And then finally, when we partitioned the FRO and did an ROH mapping, we were able to see some inbreeding patterns and shared homozygosity hotspots, especially on chromosome number 7. So our take-home message from this presentation is that indeed genomic selection for AHPN resistance is feasible and effective. And the challenge experiments we did confirm that there is heritable variation in survival, and the GWAS also confirmed that the genetic architecture for survival to AHPN is indeed highly polygenic, and that will support our genome-wide prediction over a single locus targeting or a marker selected scheme. And then finally, the population genetic analysis indeed provided us with some guardrails, which showed us that the FG and then the SG lines, they show moderate effective population size with rapid LD decay. We do have manageable inbreeding compared to other livestock species, and the ROH mapping that we did and would enable us to be able to do proper diversity management. So this is what I have done as part of my PhD. And to conclude it, I would want to talk about 1 second fun fact about shrimp. So you know how humans and cattle would have a specific chromosome, which we'll call the sex chromosome, that determines the sex of an individual. When it comes to shrimps, we do not have that. Instead, we have a single SNP, and changes in that SNP would determine if an individual shrimp is a female or a male. And that SNP is usually found on chromosome number 44. However, that SNP is also a transposon. So sometimes you'll find it on chromosome number 44. Other times it may jump and shift to another chromosome, making it really hard for us to be able to actually pinpoint the exact location of this SNP to be able to do sex differentiation. So in summary, I would like to thank USD and NIFA for sponsoring my PhD, Brenda Fragomeni for giving me the opportunity to do my PhD at UConn, all of my doctoral committee members, including Jack, Johan, Gabon, Kent, Jill, and Edward, and the shrimp improvement system. So providing us with the shrimps we use for our study, Zoetis for such an important internship that led to two published manuscripts in JDS, and then all of my lab mates in Godzilla, and also Filippo Miglio for taking a chance on me and giving me a postdoc position to begin at in January. So that is it. And thank you so much. And I welcome questions.
Thank you so much.
Okay.
It was an amazing presentation. I would like now to open the floor to any questions, either from those online or those join us in person. Okay, I can get started. Professor Flavio, go ahead.
Okay, can you hear me?
Yes,
Okay.
Yes.
I can.
Okay.
Yeah.
So I would have many questions about this work that you did. Congratulations, by the way, a lot of work for your
Thank you
PhD.
so much. Yeah.
Yeah, but I'm going to pick one or two to ask you now. But the first one that I was intrigued by the sex determination,
Yeah.
you said that it's just one SNP.
It's a
One
SNP, yes.
polymorphism, yeah.
Yes.
But how come then they would change the sex? So I bet then there is some epigenetic effects on the top of this, right? Because
Yeah.
it will not be a mutation, right? If it's determined by a single polymorphism, then should be silent by epigenetic effect, right?
Yeah. So the sex dimorphism that I spoke about you can only see that in specific shrimp species, and one of it includes like Hamata oncorhynchus. There's one species like that. When it comes to Penaeus vannamei, we do not have that. So they will start off as male and stay as male forever, and they will start off as female and stay as female forever. So the single SNP locus that I spoke about is specifically to these species and not to any other species. Yeah. You can also find the same concept in P. monodon, which we call the tiger shrimp. They also have a single locus that is able to tell you which sex you're working with, and this was recently discovered, and it's said to be on chromosome 15 as compared to what you would have in P. vannamei being found on chromosome 44. So it's very different depending on the species, and not all of the species of shrimp do exhibit sexual dimorphism, as I pointed out. There are just some of them. Yeah.
think Flavio froze.
Oh.
Yeah, let's wait for him to come back. I do have a question.
Yeah.
You mentioned that some shrimp. Oh, I think he's back.
Oh,
Oh,
sorry about that. For
oh.
somehow I was kicked off of the meeting.
Oh no.
Yeah.
But yeah. Did you hear the answer, or you want me to?
Yeah, no, I understand it's a particular species, but
Yes.
because determined just by one polymorphism, then they should have another mechanism, that usually the epigenetic mechanism, right, that would change that.
Yeah.
Like what happened with some fish, like tilapia.
Yeah.
They can change the sex, but then based on the environment, like in this case with the shock
Yeah.
of a heat shock, right?
Yeah.
But
Yeah.
I just like to ask something. Why you decide to do this? When you look at your diversity studies,
Yeah.
you did the editing of your genotypes, and you use, for instance, the math of you kept the significant math bigger than 0.05.
Yes.
Right? So
Yeah.
why did you do that? Because that would affect, for instance, the level of homozygosity, the diversity measure that we are using, right?
Yeah.
Because if these lines, they really separated, they are separated for a while. So just by, so you expect by chance that some alleles are going to go more towards fixation in one and another.
Yeah.
Those are very important alleles to define how distant they are. And you mentioned that you have the excess of heterozygosity, but you eliminate a lot of the homozygosity by doing that.
Yeah, that's a really good observation. So we actually did, so we have another results where we looked at minor allele frequency between 0 and 0.1, and we kept having like different categories of that. um for today, I was just showing the results based on minor allele frequency greater than 0.05, but we do actually have results from the other MEF categories. We did not see any changes. All we kept seeing was that the observed heterozygosity was higher than the expected heterozygosity under the different MEF thresholds that we had. But just for today's presentation, I just wanted to keep to one, and that was what I did. And also, I think you spoke a little bit about the epigenetics effects of sexual dimorphism, and I think you're right. So there's a study that was trying to look at the genetic basis for sex determination in shrimp, and they didn't find enough genetic variation to show how these shrimps are able to transform from being a male to a female later in life. And so they also said that it could be due to a lot of environmental factors. So when you talk about epigenetics, I would definitely agree with you that the environment has a huge role to play in that particular phenomena that happens for that transformation to happen. Yeah.
Okay. One more, because I think today you have a time to ask questions. So you in the first part of your study, you showed us the heritability that you estimated.
Yes.
And I assume that you showed at the liability scale, right?
Yes. It's not on the liability scale based on the way we calculated it. So we did not take into consideration the proportion of incidence that we have, and then we didn't take into consideration the height of the cumulative density of the CDF. And so because we did not,
but you use a threshold model, right?
we use the threshold model.
So the threshold model is going to give you the heritability on the line scale, right?
Yeah, but so there was a lecture that we had from Jack, and he talked about how heritabilities are calculated from a linear model and a threshold model. So yes, even though we get the variance components from the threshold model, when you look at how you calculate the heritability by taking the genetic variance over the phenotypic variance, that is what Fisher proposed, right? And that is on a linear scale. But then when we include the proportion of affected individuals in the calculation plus the height of the CDF, then that will give us the heritability on the liability scale. So that was a mistake. I would always say that, okay, the heritabilities we had here was on the liability scale, but apparently the way we calculated it is actually on the linear scale.
I would fully disagree with this.
Oh, it's okay.
Because in the linear scale, you use the phenotype as 0 and 1. That's what you're talking about?
Yes, if
you use a linear model, and that when you do that, you don't account for incidences, right?
Yes.
And the heritability will be much smaller if you have incidences that are not close to 50-50, right?
Yes. Yeah.
So I cannot agree with that. My question related with this because the two lines, they have the different mortality, so they have different incidence.
Yes.
So I would like to know what would be the heritability on the observable scale, right?
Yeah.
Because that is going to tell you how much you are going to have of variability on your phenotypes, right? And lower is the incidence. Let's say more difficult is to select against anything, right? If you have a low mortality, then it's very difficult to really select against mortality because at the observable scale, the heritability is very low, right?
Yeah. And I think
so
that's
here you see the one line. So the fast growing has higher heritability that you're showing here, but on the observer scale, because there is much, I think, lower mortality, maybe is not as
high as
exactly. Maybe they are more comparable.
Yeah.
Right. And so when you think to select for this trait, in practice, these companies, they are going to do challenges, or they are going to use the observer mortality for the selection purpose.
So usually for these companies, they would usually do a challenge experiment.
Okay.
But then the way they do it is very different from what we would actually, what the challenge would do. They would just select all families, put them in a tank, and then infect them with the disease. And then the survivors of the challenge are the ones that they would select. They would select siblings of like the survivors of those challenge, and then use them for their selection purposes. It's a bit different from what we did, but hopefully this could give them like let's say some background.
Yeah, because having this ability, because then you can create a challenge that would produce you more or less 50% of the mortality. Then you have the idea, you have the maximum of the expression of the genetic variance or the variability, right? If you use the data from mortality of the field data, then you could have a low mortality, high mortality, so the you it's more difficult to capture the genetic variability. But when you can challenge it and challenge in a way that will be like how much bacteria you put in the tank, then you can make in a way that 50% you expect that are going to die. So that you have this, let's say, case control.
Control, yes.
That will be 50%. That's the ideal for this type of trait, right? Binary trait.
Yeah, and that's why in P 2023 we had to increase the dose because we were looking at a dose that will be able to kill at least 50% of the individuals that we had in that challenge. That's the thing about like doing bacterial disease challenges that you can't control. Basically.
Okay, so I'll let other people to ask questions or comment.
Okay.
Thanks a lot. Great
You're
presentation.
welcome. Thank you.
See you in Guelph.
See you. See you in Guelph. Thank you.
Can I just add something? I think it may be interesting, depending about how those companies work. They're about 20 years behind every other species. So they're doing all the breeding by family. So they select whichever family had higher survival. But yeah, with her results, we're finally able to convince them to genotype selection candidates based on those challenge studies. So the idea is that starting, I think, next year, they will start running genomic selection for disease resistance. At least in the company we're working with. The companies don't talk much, so we don't know what is the state. But there's very few things in shrimp genomics. So hopefully with Issabelle's paper, in which I am submitting right now, that's going to get better.
Thank you. Any other questions? We do have a question in the room.
Maybe for both Issabelle and Breno, I don't know who could answer that, but this is Lucas here. I have two main ones. I think why was the window of 500 kilobase pairs chosen to be used in the study, and how many SNPs you had in average for those windows? And because you're talking about the selection of the families here, why are they so drastically different? And like, what's been like? I know, Breno, you just said that they're 20 years behind, but why are they so drastically different from each other? Like, if the purpose, I would think, for shrimp is basically the same as production of meat.
So basically, we have the slow growth line and the fast growth line. I'll first talk about the slow growth line. So in 1990, they had the first outbreak of the Taurus syndrome virus, right? And it killed a lot of, it really affected the shrimp industry. So then they decided to start what we call the specific pathogen-free shrimp lines. So the slow growth line was one of them. And the main purpose for the slow growth line was to improve resistance to TSV, WSV, and so many other diseases. So that particular line has been bred for that. And then you have the fast growth line, which is not that disease resistant. It's usually grown for the purpose of improving body weight and stuffs like that. So usually when there's an outbreak, right, in the company, what they would usually do is to go directly for the fast growth line, breed them because they grow very fast, and then sell them to their companies. And the slow growth line is just a backup, right? So those two lines have been bred selectively different from one another. One of them was targeting only disease resistance to so many diseases, and then the other one was targeting growth mainly. But now they are trying to improve the fast growth line to other diseases as well. So that's why probably they are very distinct from one another. And what was the second question? Anybody may ask?
It was just the size of the windows. Why did you choose that? But I have a follow-up on the first one.
Okay.
What is the generation interval of shrimp? Because how
It's
long
just
does it take for us to see progress of these lines and families?
so they are sexually matured around 6 months, and the entire interval is just 1 year.
Okay, and then the window size?
Oh, yeah, the window size. So we tried different windows. We tried 0.1, 1 MB, and 0.5. But then when you look at the genome size of shrimp, it's literally very small, and most of the chromosomes have very sparse markers. And when we looked at the pics that were appearing across these different window-based GWAS that we did, they were literally the same. And so we decided to just stick with 0.5 MB as our results because we saw literally the same results across different thresholds.
Okay, thank you, Issabelle.
You're welcome.
Any other questions in the room or online? I do have a question. So you mentioned that some shrimp species, they can change the sex during the lifetime. And I was wondering, how is this handled in our genetic models? Do we treat this as the sex as a fixed, or do we account for the change of over time?
I know that recently there's no model that accounts for sex-specific effects in their models because of this whole thing that has to do with their sexual dimorphism. Usually when it comes to like cattle evaluation, we would exclude the sex chromosomes, right? But because basically sex is being determined by a single locus, and this locus is so movable. It's a transposon, basically. It can jump from one place to the other. It's just hard to locate and then get rid of it. So it's included in the entire evaluation. That is what I know. But we keep improving. There are more technologies coming up, and just recently there was a paper that was released on the tiger shrimp, and it was very specific to locate the locus on chromosome 15, and apparently that one is not a transposon. So maybe we can start progress from there and then see how we can work with the transposons later on. But yeah, that's all I can say with respect to that.
That's perfect. Thank you. And you also mentioned that it has a difference in the chromosome 7. I was wondering if you have like looked on that, on how they are different.
So you mean the enrichment we did, right? This
It
one.
was the last part. Yes.
Yeah. So I did take a look at that position on chromosome 7, and there are so many genes in there that I can't actually pinpoint which gene, is like very important at this point. So what we've been thinking about is doing a selective sweep analysis and then see what kind of markers are in LD, or like what kind of homozygous segments we can find, and then probably we can take it from there. But I think I'm done with my PhD for now, and so I'm not. I don't know. I don't think I'll be doing it. Probably Breno might find someone else to do it. For now. Yeah.
Okay. Thank you.
Well,
Any other questions in the room or online? Yeah, we do have a question in the room.
Mine's working. Oh, okay. Sorry. Hey, Isabel. I just had a clarification question on, you mentioned sexual dimorphism in shrimp,
Yeah.
but then you reference the switching of sexes between shrimp. And I think aren't they two different things? So sexual dimorphism, as far as I understand, is the differences in size or behavior, etc., between males and females, while I believe it's called sequential hermaphroditism is when they switch sexes. So
Yeah.
basically my question is, do all shrimp exhibit sexual dimorphism and sequential hermaphroditism, or some do and some don't?
So for shrimp, it's called platodic
Yeah.
hermaphrodite. Oh my God, that's a long name. I don't want to
Yeah.
go into that. But basically, so they do not lose the ability to be males. They gain the ability to be females as well. I don't know
Okay.
if you get, yeah. So in that part, they do not like forever lose the ability to be. So they start off as males, and then as they grow, they gain an extra ability to be females as well. So they can produce eggs and sperm at the same time. And so that is where you have the hermaphroditism coming in.
I
Okay.
see.
And then when you
Okay.
come to P. vannamei, it's not the same. With P. vannamei, they start off as whoever they are, and they just stay that way forever.
Okay.
Yeah,
But
so
all
it's
shrimp
really
species.
species different. It's very
Gotcha.
species dependent.
Right. So that's the hermaphroditism aspect. But for the dimorphism aspect, all of the species exhibit that, or only a few?
Only a few, not all of them.
Okay.
Yeah.
Okay.
Only a few.
Gotcha.
Yeah.
Thank you.
You're welcome.
Any other questions? We do have a question. This will be the last one. Just 1 second.
Oh, hi, Isabel. Just a quick question. You say they use like a fast growth line and a slow growth. Any other name of this specific line, or it's just fast and slow?
Could you take the question again?
Yes,
Could you
you
repeat?
mentioned that you use a slow growth and a fast growth line. Any other specific name for this line?
No, that's the name that SIS provided to us. So basically, it's SIS fast growth, SIS slow growth. That's how they are named in the company. I don't know any other name for them.
Okay, thank you.
You're welcome.
This has been a great discussion. Thank you again for presenting to us, Dr. Issabelle. Thank you everyone for coming. This CGIL seminar will be posted on YouTube, and I will share more details about that later. See you all next week. Have a nice weekend.
Thank you so
Bye-bye.
much. Bye. Bye.